So we've got Sharon Tamir hot on the heels from Tom Dye's talk to carry on what he was talking about in humans to what Sharon you're finding about in mice, right? Sharon is an M. No, we'll try to stick closer to time, but I wanted to take 2 seconds to say that Sharon, MD, PhD student in Cincinnati, working in John Hoganish's lab, and she's studying sleep and circadian rhythm disturbances in SHINE Syndrome using a mouse model. And she, I think you also, yeah, come on up. You also co-authored the paper that Tom referred to, that he'll send out for us to read.
Yeah,
So,
sure.
okay, so I bow down. Thank you for coming.
Thank you. Thank you guys so much for having me, and particularly to the DLG4 SHINE board for putting all this together. We're really, really excited to be here. Now, I think this is what it is. Big green button, excellent. Okay, so yeah, my name is Sharon Tamir, and today I'm really excited to talk to you guys about a patient-derived mouse model of SHINE syndrome that our lab developed. So first, super big picture overview. As biomedical researchers, our goal is really to understand the manifestation of disease, right? What causes it, how does it manifest, and to look at and develop treatments, if possible, for our disease, right? And the way we do this is by, as much as possible, using what we learn from our patients, tests in our patients, to do this. But whenever we can't do that, if there's a procedure that's maybe too invasive or not the best for our patients, we try to do that in mice, right? So these mouse models really helps us understand what's going on with these manifestations of the disease in addition to what we learn from the patients, and to test these possible treatments before we put them in our patients. So our lab was really interested in making a SHINE mouse, right, in order to model what we see in a patient at Cincinnati Children's that has a particularly severe form of SHINE syndrome. The way we do this is we use CRISPR-Cas9, which I'm sure many of you have heard of. This is a means of very specifically inserting our gene with the variant of interest into the mouse, so it's expressed ubiquitously in the mouse, like we see in the patient. And that's what's depicted here on the screen. So we're going to take our Cas9 system and the variant of DLG4 that we want to insert. We actually insert that into the fertilized egg of a mouse, and then we breed that up into a mouse colony. This takes a year or more, so this is a very long process from beginning the process to actually being able to start studying the mouse. Once we have a mouse model, we want to evaluate the mouse, right? Mice are not people. So we want to see to what degree is what we see in the patient recapitulated in the mouse. And this tells us what can we actually learn from this mouse, what can it inform us about what we see in the patient specifically, right? So what characteristics are observed in our mouse model? And in order to start asking that question, we need to see what do we see in our patient, right? What are we comparing it to? So here's what we see in our patient, right? Our patient has expresses the variant gene, right? Our patient has decreased PSD95 protein expression. Our patient experiences sleep disturbances, has intellectual disability and neurological disorders, and severe epilepsy. And so all these things are things that we want to evaluate in the mouse model to see, are these aspects that we can actually study in this mouse? So first, we'll talk about gene expression. You guys have talked about this a lot today, so I'm just going to do a super quick review so I can tell you how we actually measure this in the lab. So just like Dr. Tamir and others talked about earlier today, our genes are expressed in these transcripts called RNA, and we can measure how much of this RNA is expressed in our cells. So what we do is we'll take some tissue, in this case it's going to be mouse brain tissue, and we're going to isolate that RNA. And then we're going to use this process called the polymerase chain reaction. This allows us to specifically amplify our gene of interest. In this case, it's going to be DLG4. And then we use this process called gel electrophoresis, which allows us to visualize if there is or is not gene expression, right? So these sort of bands are going to be what tells us, yes, there is DLG4 gene expression here. All right, so now let's look at the data. Let's start with what we see here on the left. Right, so this is looking at wild type or healthy DLG4 gene expression. In the first column, you see what we call a ladder. This tells us the approximate size of the RNA or the transcript that we're looking at. Here, I amplified a transcript that's about 300 base pairs. So that's what we're looking at there. In the next two columns, you see the wild type or the healthy DLG4 mouse, right? So this is a mouse with 2 healthy copies of DLG4. And then in the next 2 columns, you see the SHINE mouse. This is a mouse that has 1 healthy copy of DLG4 and 1 variant copy of DLG4, the same variant that we see in our patient. And what you can see here is that both the wild type mouse and the SHINE mouse express the healthy DLG4 gene. So that's what we're seeing here on the left. On the right, we're looking at variant DLG4 expression, right? So again, we have our ladder, just like before, but this time we don't see any gene expression in the wild-type mouse. And this is what we expect, because the wild-type mouse does not have the variant. So this is good. But we see that the variant is expressed in the SHINE mouse. Now, thanks to collaboration with the tumor lab, we know that the patient also has expression of the variant. So we see this is a way in which the mouse is recapitulating what we see in the patient. Now, next we wanted to look at protein expression. And the way we do this is through this method called the Western blot. You might have heard of this before. This is very commonly used. And just like we talked about earlier today, genes are expressed as these transcripts, RNA, and then these RNA are reconfigured, produced into protein. And the protein is really the functional element of the cell, right? This is the one that's traveling to the synapse, that has that important scaffolding function that is sometimes lost in DLG4 SHINE. The way we assess the expression of protein is through this process. So we're going to, again, take our tissue. In this case, again, it's going to be brain tissue. We're going to isolate the proteins out of it, and we're going to run it again on a gel electrophoresis. This sort of spreads out the protein based on size, where bigger proteins are near the top, smaller proteins get closer to the bottom, and then we transfer this result, this protein spread, onto a membrane, and this membrane is often called a blot, hence the name Western blot. This membrane is then going to be incubated with antibodies. So here, let me move this a little bit. So this sort of yellow kind of picture on the bottom, this is a representation of the membrane again. The red dot is our protein of interest, right? This is our DLG, our PSD95 protein. The first antibody is specific to that PSD95 protein. And the second antibody binding the first antibody, so the blue one binding the yellow, this allows us to visualize that protein. So in this way, we can specifically visualize how much of our protein of interest there is. And then that's what we see here on the blot. This is our final result, right? Where the big, darker, thicker bands mean higher protein expression. There's more of it, the antibody bound to more protein. And the smaller, thinner, lighter bands, that means less protein was expressed. Okay, so now let's look at the data. So here we see again a ladder. The ladder tells us the approximate size of the proteins that we're looking at. That's our first column. Our second column is for our wild type or healthy mouse, and the third column is the SHINE mouse that has 1 healthy copy and 1 variant copy. So we can see that the PSD95 band is just under 100 kilodaltons, and that's why it's called PSD95, because it has a molecular weight of 95, and that's what's sort of demonstrated here, right? It's just under the 100 kilodalton band. So we can see already that the wild-type mouse band looks a lot darker and bigger than the SHINE mouse band, right? Suggesting that there is a higher expression of PSD95 in the wild-type mouse and a lower expression in the SHINE mouse. Now, what is the second band? So this band underneath, this is something called a loading control, and you'll see this a lot if you're looking at papers that use Western blots. The loading control is making sure that we're putting the right or a similar amount of protein in both of these columns. Because you can imagine, if I put 2 times as much sample in the first column as I did the second, it would look like there's 2 times as much protein, right? But there wouldn't actually be a real difference between them because I just messed up the sample. So this gives us something to normalize to, right? We have a protein that we know is ubiquitously expressed in all of these conditions. And we can use that as a reference to make sure that the comparisons we're making are real comparisons. And that's what we have quantified here. So we can see that the wild-type normalized amount of PSD95 is much higher than what we see in the SHINE mouse, similar to what we see in the patient. Now, our lab is a sleep and circadian rhythms lab, so we're particularly interested in understanding sleep disturbances in our patients and, by proxy, in the mice. The way we do this is by using the system called the piezo sleep system. This is a non-invasive way of measuring sleep in the mice, and the way it works is the mice have their cages, normal sort of home environment, with this very, very sensitive pad at the bottom that detects their breathing. So awake mice, as you can see, have this very chaotic sort of detection, right? They're moving, they're breathing at irregular rhythms, maybe they're eating, maybe they're drinking, they're moving around. Sleeping mice have a very regular 4 Hertz rhythm that we can detect, and in that way actually discriminate between sleep and wake. So what we found when we were looking at sleep in the SHINE mouse was first and foremost that there was no real difference in the percent of time they spend asleep during the resting phase. So the total sleep time was the same. We also found that the number of sleep bouts the mice have, right? How many times they fall asleep and wake up and fall asleep and wake up, which is very normal for mice to do that multiple times, seemed to be the same between our wild type and our SHINE mice. But interestingly, when we look at the proportion of short sleep bouts to the total number of sleep bouts, we found that the SHINE mice have more short sleep bouts. And we also found that the SHINE mice have a longer average sleep bout length. So what sort of picture does that paint? So I'm going to give you a very rough sort of schematic here of what we're looking at. So let's pretend that the green here is sort of representing our wild type sleep, and the blue is representing our SHINE mouse, particularly in the male's sleep. The length of this bar is going to represent the total time of sleep, and you can see it's the same between the two mice. And the space between these white barriers, we're going to call that one of the sleep bouts, right? One of the times they stay asleep and then they wake up for a bit and then they stay asleep again. We see that the SHINE mice have more shorter sleep bouts and then these compensatory longer sleep bouts, right? And what does that mean for their sleep or their quality of sleep? It's possible that these mice are probably not getting to the REM stage as much as their wild-type counterparts, so decreased REM sleep. And we find this to be particularly interesting because we know that our patient has somewhere around a 50% decreased REM sleep compared to what is expected in his sleep. Of course, we're also really interested in looking at intellectual disability in these mice, and now where we can't measure that directly in mice, we can measure learning and memory through this test called the Morris Water Maze. This is a test where you get this large bucket of water, and you paint the entire bucket black, and you also have a platform on there that's also painted black, so it's very hard to see. And the mice are trained to be dropped somewhere in the swimming pool and to find the platform. And if they are capable of learning this process and remembering where the platform is, they will perform better by having a more efficient path to the platform, right? So we can see here, like in the dotted line, that's a pretty efficient path to the platform, where the straight line is a very inefficient path to the platform. So higher path efficiency means a better ability to learn and remember. And when we look at the data, we see that our lab did a 5-day trial of this test on our wild type and our SHINE mice, and we see that consistently across the board, the SHINE mice perform worse, have potentially impaired learning and memory compared to the wild type mice. Neurological disorders could also be difficult to test in mice, so one of the ways that we try to approach it is by testing these sensory motor reflexes. And there are 2 tests for this. The first one depicted here is called the acoustic startle reflex, and it's very much what it sounds like. The mice are exposed to a noise, and that noise travels through our auditory pathway, right, which is very much sort of in our brain area, and then from there to our spinal cord and to the rest of us in order to elicit this startle response, right? So this tests kind of the ability for a central processing and responding. There's also a tactile reflex, where this is instead of a noise, you have a puff of air. And this puff of air elicits a response in the peripheral nervous system that travels through the spinal cord and back to the peripheral nervous system to elicit this response. So we have a central system and a more peripheral system way of looking at neurological processing. When we do these tests in our SHINE mice, we have some really interesting findings. We see that on acoustic startle, right, this is this more central through the brain process, the SHINE mice have a stronger augmented response. But when we look at tactile startle, the SHINE mice have a damped or impaired response, suggesting that PSD95's role could be increasing sensitivity in the central nervous system and damping sensitivity maybe in the peripheral nervous system. And we know that our patient experiences some sensory motor abnormalities as well. Lastly, of course, we were really interested in seeing if our mice recapitulate the seizure symptoms we see in our patient. And the way we do this is by collaborating with a lab who completely specializes in this, free-roaming EEG in mice. The Danzer lab, they're incredible, at Cincinnati Children's. And so we take our mice and we insert the EEG detectors directly into the brain and then the transmitter just underneath the skin so that the mice don't have to be in one place for the entire time. They can walk around their cage, eat, drink, sleep like normal, and we can detect if there are any spontaneous seizures. So this allows for some really nice, continuous, non-invasive recording. And we did this recording for 2 weeks and found no spontaneous seizures, which you can imagine was very disheartening. But when we really thought about it, we thought life is not this stimuli-free environment like it is for the mice in the cage. And this is where your guys' perspectives would actually be really helpful. What kind of stimuli induce seizures in your kids? If you can talk to me about this later, I would really appreciate it, and that would help us better understand how we could further test these mice to see if maybe with certain stimuli, with certain stressors, maybe these mice are actually more seizure prone and we just don't see it yet. So that's where we're at with evaluating our mouse models so far. I'm just going to do a very quick review and show you how we were able to really recapitulate a lot of the symptoms we see in our patient in our mouse. So we know that our patient has aberrant gene expression of the variant, and we see the same thing in the mouse. When we look at protein expression in the patient, we see that's decreased, and we also see decreased PSD95 in our mouse. While our patient experiences sleep disturbances, our mouse has this interesting variable sleep out length phenotype that could be affecting their sleep architecture. We see that our mice has impaired learning and memory and abnormal sensory motor reflexes compared to the healthy mice. And currently we see no spontaneous seizures in our mice. So big thank you so much to the DLG4 SHINE Foundation, to the board members, to all of our collaborators and lab members, and absolutely to the patient and his family that are working with us. Thank you.